Question 6. Question 6 just has you go through, we've seen this one before, just labeling the gene model. Hopefully this was all pretty straightforward for you. Let me make it a little bit smaller so my head's not in the way. I don't think you need to see my head, but I don't know. Y'all kind of seem to enjoy it. I don't know. It makes it more personable, right? Anyways, we have our gene model here just to kind of orient yourself. We've got a sequence starting somewhere out here. We can't see the edge. It's coming in. There is an enhancer region upstream of our eventual gene. We call this the distal control elements. In a eukaryotic cell, this bending over of the DNA is actually required to bring that enhancer into close proximity to the promoter region where the polymerase will actually end up binding. And so then downstream of that, so in towards the 3' end, we then encounter the gene. So that should get you oriented so far. Let's go through these and name them. So one's not my favorite, but there's only so many options. I believe in this one it's just worded the DNA bending protein. I don't think anyone misses that one. That one's pretty obvious if you're even just reading things. It's a complex of proteins that helps the DNA get in this correct configuration. It does require that the sequence in here be of a certain length, have certain patterns in order to work, not have certain patterns, not have like a big old histone glomped onto there. There's a lot of stuff that you can do to this region of the DNA that actually prevents downstream transcription. And so, I mean, it is something to pay attention to. In my experience, I haven't seen a lot and I don't do a lot of this type of research, but this I think is just so hard to predict. I don't see a ton of research on it. Hopefully that changes in the near future. And for the record, there's some biologists right now that just started screaming and they don't know why, because that's probably their life's work of illuminating this. It's just that I think that for those of us who are involved in trying to use reference genomes downstream, there's so much work that goes into annotating these genomes and trying to understand these super complicated structures. And that's really more where I exist within the genetics field. We don't have the resolution to look at that because these are all just so kind of chaotic from our point of view. Molecular biologists, like I said, have a different point of view. All right, let's keep going. I'm just really putting myself into a corner here. So down here we have some sort of like kind of umbrella term for this whole region. We can see that there's one here that I've kind of partially left defined, so that way you didn't call all of this the distal control elements, which is what that 6 is covering up. Because 2 is trying to encompass this whole region. So we call this whole region of DNA the enhancer. The enhancer is complicated and there's parts to it and things bind to it and interact with it. But the region of DNA that does all of those interactions and gets interacted with is called the enhancer. Number 3, it could have been probably a few different things: transcription factors, mediators, probably some other things I'm not thinking of, but it's going to be something along those lines. And then in comparison, 4 is going to be something that specifically interacts with the enhancer region. So again, there's some different things like you could have told me here. It could have been like repressor or activator, but it needed to be one of the compounds that actually interacts with the enhancer. So that's kind of the specific thing that needs to go here. And then finally, the 5, that's going to be either promoter or untranslated 5' region or something along those lines. And if you had those as different options, you'd probably need to get some more clarification. So let's go see what our choices are. So number one was that DNA bending protein. Number 2 was the enhancer. I think we're probably going to see a good number of students who put operon for this, and operon makes sense, although operons tend to be this whole sequence down here in prokaryotes, where we don't get this whole bending process. So by this bending process being present, we're identifying that this is in fact a eukaryote and using a eukaryotic type gene regulation system, and so not an operon, an enhancer. Number 3 was that mixture of different things. Let's look through here. Not a histone. Only thing that makes sense is transcription factors. You would maybe say activators. I think that would be a reasonable, except activators act on the enhancer. And so 4 is definitely going to be the better choice for activator. So you get to 4, you do activator, and then you get to 5. And 5 is going to be our promoter. If I had had a 5' untranslated region, maybe that one. And then finally 6 was the distal control elements. Down here is the proximal control elements. So this region down here. So distal being more distant, proximal having a greater proximity. If those help you, I don't know. They would help me, but I don't know if they'd help you. So anyways, that would be the distal. That's it.